Dear students, welcome to our biology program in Egyptian TV. Today we are going to complete the DNA structure. In the last session we talked about the DNA structure and how the DNA is consist of number one, ribose sugar, also nucleotide, okay, and this is the structure of DNA, consists of deoxyribose sugar, nitrogenous base, and phosphate group. Also, we talked about the structure of the bacterial cell and how the bacterial cell, it is the simplest cell which contain only one cell. And inside the bacterial cell, there is what is named by plasmids. Plasmid is the DNA inside the bacterial cell, which consists of the ring DNA. Let us to see what is the structure of plasmid and also what is meant by plasmid. Okay, plasmids are much smaller and circular DNA molecules that are not complexed with proteins. Means that the plasmid is a circular DNA and not joined with protein found in the prokaryotics. موجود في البروkaryotics. Okay. What is the site of plasmid? Sites means the position of plasmid. Where the plasmid is found? Found in two cells. Number one, prokaryote cells means some bacterial cell which contain one or more plasmids. Prokaryotics, they are simplest type of cell. And also found in eukaryotes, eukaryotes like the yeast fungus. Not all types of eukaryotics contain the plasmid. Now, the sites of plasmids found in two cells. Number one, prokaryotes. Number two, eukaryotes. Prokaryotes, some bacterial cells which contain one or more plasmids. Eukaryote, like the yeast fungus, which also found inside it plasmids. Okay. What is the importance of plasmid? Why the plasmid is necessary or the plasmid must be found inside the cell? First, plasmids are widely used in the field of genetic engineering. Means, if you talked about the genetic engineering or the genes, you must describe the DNA, and the plasmid is the simplest form of DNA structure, where the bacterial cell replicates any plasmid inside it during replication of its main DNA. Okay, now we are describe what is the plasmid again. It is the genetic engineering or the DNA structure. Okay, and the scientists take the advantage of this activity by introducing artificial plasmids into the bacterial cells in order to obtain several copies of the plasmids. Second, DNA in eukaryotes. Eukaryotes, like what? Eukaryotes, like the higher plant, human, some living organism which contains the genetic material surrounded by nuclear membrane. Okay? Must be the DNA found inside the cell which contain nucleus, and this nucleus surrounded by the nuclear membrane. The nuclear membrane separates the DNA and separates it from cytoplasm, and DNA is organized into several chromosomes. The human somatic cells contains 46 chromosomes. Chromosomes appear in eukaryotic during the cell division. Okay, in the last session, we talked about the human and the structure of DNA. You know that the human cell contains nucleus, and inside the nucleus, there are the DNA structure. Deoxyribonucleic acid. This DNA contains fixed number for the chromosome which characterize each species of living organism. Like first the human. Human contain 46 chromosomes, while onion plant contain 20 chromosomes. So each one of the species for certain living organism contain fixed number of chromosomes. These chromosomes found inside the DNA structure, which is found in the nucleus. Okay, the nucleus surrounded by nuclear membrane. Okay, now what is the structure of nucleus? To know the structure of nucleus, we studied in the last session that the nucleus consists of 4 structures or 4 4 تركيبات موجودين في النيوكليوس عندي. نمبر 1 nuclear envelope أو nuclear membrane. نمبر 2 عندي nucleolus. نمبر 3 nucleoplasm. نمبر 4 chromatin reticulum. Okay, nuclear membrane separates the component of the nucleus from the cytoplasm. Nucleoplasm, it is a fluid which contains nucleolus and chromatin. What is the nucleolus? Nucleolus, it is responsible for the synthesis of protein to make hormones and other nutrients for the nucleus. The last one, what is the chromatin reticulum? Chromatin reticulum, it is the material, بنقول عليها minute tangled filaments. minute tangled filaments, خيوط صغيرة متشابكة, which form the chromosomes. Okay, this is the chromatin reticulum, which contain the DNA or the genetic engineering or the chromatin reticulum. Okay, now we are going to see what is the membrane which separates the cytoplasm and DNA. Okay, the structure of chromosome. Each chromosome contains a single DNA extending from one end of the chromosome to the other end. Okay. Also, in the last year, we talked about the structure of chromosome, and we said that each chromosome consists of 2 chromatids joined by centromere, and each chromatid consists of DNA. Okay, and this DNA coiled around a protein named by histone. DNA molecule is coiled and folded many times and associated with various proteins. This protein called histone, which forming the chromatin, which contain roughly equal amount of DNA and protein. Okay, what is the chromatin? Chromatin, one molecule of DNA, means that one strand of DNA consists of, or one molecule of DNA means one nucleotide which contains 4 nitrogenous bases extending from one end of the chromosome to coiled and folded many times and associated with protein. You know what is meant by folded? Folded means make many يعني متنيين يا ولاد كده علشان to take small area. معنى كلمة folded معناها إن هي become small in area to take small space inside the cell. Okay, what is the histone proteins? Histones, they are well-defined group of small structured protein that present in great amount in the chromatin of any cell. Okay, where they have a high content of the 2 basic amino acids, the 2 amino acids arginine and lysine. Okay, let us to describe what is the types of amino acids. What is the amino acids? Amino acids is a type of protein. Means what is the protein? Protein is a polymer macromolecule which enters the structure of the muscles, the hair, ligaments, tendons. Any part of your body need to grow, need protein. Okay, what is the building unit of protein? You notice that the building unit of protein named by amino acids. Glycine, arginine, lysine, this is the types of amino acid which join together to form a certain type of protein which enters the structure of DNA or the structure of the cell. Okay, okay. Now we talked about lysine, arginine, which enter the structure of protein, which enter in the structure of DNA formation. Okay, there are another types of amino acids found in your body like histone, glycine, valine, alanine. They are different types of protein or amino acids which enter the structure of protein needed for many body organs inside your body. Okay, now we are going to what is the histone. Histone is a type of protein bind strongly with the negatively charged phosphate group. that are present in DNA molecule. You know that the DNA consists of nucleotides. This nucleotide consists of 5 carbon atoms. Carbon number 1 join with nitrogenous base, while carbon number 5 join with phosphate group. Because the terminal alkyl group are of the 2 amino acids, arginine and lysine, has a positive charge at the normal pH of the cell. You know that is the normal pH of the cell means that is the cell contain alkaline medium. Alkaline medium means that is the pH greater than 7. أكتر من 7 يا ولاد. ده البي اتش. لما البي اتش تبقى أكتر من 7 يبقى ده معناه إن أنا عندي الألكايل جروب أو التيرمينال ألكايل جروب أرجينين ولايسين عندهم البوزيتيف تشارج بتاعتهم جوين مع البي اتش أو normal to the pH. Okay, what is the non-histone protein? Okay, they are heterogeneous group. What is meant by heterogeneous? Heterogeneous means different group, and this different group differ in structure and regulatory protein. And this non-histone protein has many functions that found in the structure of chromatin. Okay, non-histone protein have many different functions because they contain number 1 structure protein that enter the structure of definite part of DNA molecule and play an important role in special organization of DNA within the nucleus. What is the structure protein? The structure protein, again, this is a main or contain or organized the DNA within the nucleus. What is the regulatory protein? The regulatory proteins determine whether the DNA code will be used in making RNA protein and also enzymes or not. Regulatory proteins معناه يا اولاد اللي هو البروتين المنظم. هو ده اللي هيقول لي اذا كان هقدر اعمل دي ان ايه كود شفره للدي ان ايه باستخدام الار ان ايه. يعني ايه؟ أنا عارفة you know that is the structure of DNA or the DNA structure to mean this transcribed يعني يستنسخ أثناء ما يحصل cell division يا ولاد. اللي بيحصل في cell division أو انقسام الخلية بيحصل إن ال DNA بيحصل له transcribed يعني بيستنسخ منه. نخرج منه إيه من ال DNA؟ بنخرج منه حاجة اسمها ال RNA. طب وخدنا قبل كده مين هم الانزيمز اللي بتساعد في كده؟ اللايجيز والبوليميريز وكل الانزيمز دي. طب الدي ان ايه ده عباره عن دايوكسي نيوكليك اسيد. طب مين هو الار ان ايه؟ ده بنقول عليه رايبو نيوكليك اسيد. رايبو نيوكليك ايه اسيد. احنا عارفين ان الدي ان ايه عباره عن دبل هيليكس او 2 ستراندز، لكن الار ان ايه ده عباره عن سنجل ستراند وده دراع واحد بس منفرد. وعارفه ان الار ان ايه ده بيختلف عن الدي ان ايه في النيتروجينس بيزز. عندي النيتروجينس بيزز بتاعتي اللي موجوده في الدي ان ايه هي الثايمين والسيتوسين والجوانين الثايمين وادينين وسيتوسين وجوانين. While the nitrogenous bases found in RNA uracil, okay, instead of thymine, adenine, cytosine, and also a guanine. So the DNA to transcribed عشان نستنسخ من ال DNA RNA علشان يقدر يبتدي يعمل السيل ديفيجن أو ينقسم جوه السيل. فدوت بنقول عليه اسمه regulatory proteins. Okay. هنا what is meant by packing of DNA. معنى كلمة packing of DNA if we imagine that the DNA double helix لو أنا عارفة إن ال DNA double helix of each chromosome lined up and stretched out it would be about 2 meters in length. Therefore طبعا يا جماعة هو هنا بيقول لي ان الدي ان ايه موجوده عندي في الايوكاريوتيك سيلز مش موجوده على هيئه مش ستريتشد يعني مش مفرود، لانه لو اتفرد هيبقى طوله في حدود 2 متر. لكن ايه اللي بيجرى؟ بيجرى ان بيحصل له حاجه اسمها باكينج. باكينج يعني هو متجمع او فولدد او متني كله مع بعضه to take a small area inside the cell. Okay. The histone, it is a type of protein which enters the structure of DNA. The histone and other proteins are responsible for packing these long molecules. يعني اللي مسؤول عن عملية ان يبقى ال DNA عامل باكينج او عامل سيركل او ان هو folded inside the cell، اللي مسؤول عن كده الهيستون ومعاه other two proteins. اوكي، to fit into the cells عشان يقدر يدخل جوه السيل نيوكليوس، وده بيبقى من 2 to 3 مايكرون in diameters. يبقى كده معناه ان انا خدت سبيس قليل جدا في السيل من 2 to 3 مايكرون في الدايمتر بتاعها. You know what is meant by diameter؟ diameter ده معناه ان انا لو عندي سيركل، عندي دايره، الدايره دي في من التو بولز بتوعها اللي هو الخط اللي بيعدي بالبرنسبل اكسس بتاعها، ده اسمه diameter. الدي ان ايه بياخد من 2 to 3 مايكرون ان diameter. فطبعا احنا شايفين اهو الدي ان ايه ده الاستراكشر بتاعه دبل هيليكس زي ما انا شايفه، وهو فولدد مع بعضه، ولما بيبقى فولدد بيعمل باكج علشان يبقى يدخل في الاستراكشر بتاع الكروموسوم. دايما انا شايفه ان الكروموسوم بيتكون من 2 كروماتيدز، each كروماتيد عباره عن ستراندز من الدي ان ايه. اوكي، يبقى ال DNA molecule is wounded. What is meant by wounded؟ يعني twisted or coiled around each other. Okay, wounded around the clusters. Clusters means يعني groups يا ولاد. Groups مجموعات من الهيستون بروتين. Okay, these groups of histone which around in the DNA forming a string. This string is called the nucleosome. This string is called what? Nucleosome. Okay. This shortens of the DNA molecule about 10-fold. 10-fold means ان هو متني عشرات المرات يا اولاد علشان يبقى باكج او يحصل له باكد، يعني يبقى جروبد كده او مقفول about 100,000 times more tightly to fit into the nucleus. Okay. The string of nucleosomes is coiled again to pack the nucleosome together. However, even this is not sufficient to shorten the DNA molecule to the required length. The tightly coiled string of nucleosomes are arranged in large loops that are held together by the structure non-histone proteins to form the chromatin. The chromatin is packed up as tightly as possible to be condensed into the chromosomes. What is the nucleosomes? Nucleosomes, they are a string of particles found in the chromosomes and consist of DNA molecule wounded or wrapped around the clusters of histones to shorten the DNA molecule about 10-fold. Okay, now what is the DNA? The DNA, like we described in the last slide, the DNA is the smallest organ or smallest particle found inside the cell, and this DNA become folded and make many tendons around each other to form the structure. So DNA is folded inside the nucleus of the cell to make a nucleoplasm. عشان يبقى عندي يا اولاد nucleosomes ونيوكلوبلازم موجودين جوه النيوكليوس. اوكي، انا عارفه تاني هعيد الحته دي. جوه النيوكليوس بنقول ان انا عندي في الاستراكشر اوف نيوكليوس في عندي نيوكليير انفلوب، عندي نيوكلوبلازم، عندي حاجه اسمها الكروماتين ريتيكولم، وعندي حاجه اسمها نيوكليولس. تمام. جوه في الكروماتين ريتيكولم بتعمل الكروموسومز، وقلنا ان ايتش كروموسوم كونسيست اوف دي ان ايه. طب الدي ان ايه دوت لو عملت له ستريتشد هلاقيه 2 متر. طب انا مش عايزاه 2 متر، هو ربنا خلقه جوانا انه حصل له حاجه اسمها 10 فولدد. 10 فولدد يعني متني عشرات المرات. ليه بقى؟ تو تيك سمول اريا. وبعدين غير ان هو تيك سمول اريا، بقول ان هو اللي بيساعدني في كده ان الرينجز بتاعت الهيستون بروتين بتساعد ان الدي ان ايه يعمل كويلينج علشان تو فيت سمول سبيس. What is the genome? Genome is the total of all genes, all DNA that are found in the cell. The cell contains DNA which carries the genes, which carries the instructions needed to construct the cells, and also sequence of nucleotide. يعني ايه برضو sequence of nucleotide that is responsible for making a protein compound. This means that is the we must to know that the protein which needed to form the hair. لو انا عندي بروتين مسؤول عن تكوين الشعر بتاعي، تمام، او الكالر بتاع السكين بتاعي، او الكالر بتاع الاي ايرس بتاعتي اللي هو لون العين، مين اللي مسؤول عن التكوين ده؟ نوع من البروتين. طب البروتين ده علشان يتكون محتاج ايه؟ لازم يبقى عندي حاجه اسمها sequence of nucleotide. sequence of nucleotides means ان في ترتيب للنيوكليوتايدز هو دوت اللي بيديني البروتين which responsible for formation the compound. This sequence of nucleotide from which the ribosomal RNA, tRNA, which enter in the building of ribosomes is transcribed. Sequence of nucleotide from which transfer RNA اللي هو ال eRNA which carry the amino acid during protein synthesis is transcribed. You know what is meant by transcribed? Transcribed أو عملية الترانسكريبشن يا ولاد. يعني إيه كلمة transcription؟ transcriptions معناها إن أنا بستنسخ حاجة، يبقى في تاني حاجة اسمها transcription. transcription معناها إن أنا بستنسخ. إزاي يكون عندي DNA molecule وأطلع منه RNA؟ عملية الترانسكريبشن دي علشان ال RNA يبتدي يعمل formation of certain type of protein. مش هيعمله جوه النيوكليوس بتاعت السيل، لا ده جو اوتسايد، بيخرج بره النيوكليوس ويروح عند السيتوبلازم. ده during cell division. يبقى خلي بالي قوي ان اثناء السيل division what happens during cell division? The DNA transcribed from each RNA. This RNA go outside from the cell or go outside from the cell nucleus to the cytoplasm according أو through nuclear pore. طب what happens when the RNA go outside the cell? When the RNA go outside or the ribosomal RNA go outside the cell, it responsible for the synthesis of protein. مسؤول عن تخليق البروتين اللي موجود في السيتوبلازم بتاع السيل. يبقى أنا عندي نوعين من ال DNA موجودين عندي بيحصل فيهم سينسايز للبروتين. نوع جوه النيوكليوس نفسها فده الدي ان ايه الاساسي بتاعي. طب لما الدي ان ايه الاساسي يعمل ترانسكريبشن ويستنسخ من نفسه ار ان ايه، it will go outside of the nucleus through the nuclear pores to the cytoplasm of the cell to formation the protein inside the cytoplasm. طب what is the function of this protein? The protein inside the cytoplasm responsible for number one formation of enzymes. زي ما انا عارفه ان الانزيمات اللي موجوده في جسمي محتاجه للبروتين. بعد ما بيعمل البروتين ده، بعد ما بيعمل الانزيم البروتين ده هيعمل بيه الانزيمز. طب والانزيمز دي غير الانزيم هيعمل ايه؟ هيعمل تو فانكشنز اجين. مين هم؟ حاجه اسمها growth وحاجه اسمها regeneration. growth يعني النمو وريجينيشن يعني التجدد. يعني ايه نمو؟ يعني ستيل عايزه تكبر، عايزه تحصل لها تجيب رايبوسومز وتجيب بروتين عشان تو بي لارج ان سايز، يبقى اللي مسؤول عنها الار ان اي. طب افرضي ستيل عايزه تعمل ريجينيريشن. يعني ايه ريجينيريشن؟ يعني عندي الميتوكوندريا اللي موجوده مثلا حصل فيها دامج، او وات ايفر عندي مثلا البارت من الجولجي بادي مش بتقوم بالفانكشن بتاعتها زي ترانسبورتنج فيزيكلز. تعمل ايه؟ يقوم الار ان ايه اللي عمل البروتين ده هو اللي responsible for formation this or regeneration this organelles found inside the cell. The genes in prokaryotes, the genes that are responsible for the RNA and the proteins synthesized represent most of the genome. Genome قلنا الديسكرايب بتاع genome. Okay. The genes in eukaryotic. What is the genes? less than 70%، أقل من 70% من الجينوم serve the function of RNA. يعني أقل من 70% من الجينوم بتخدم وظيفة ال RNA، يعني بتساعد بالعربي كده في إن ال RNA يعمل تخليق للبروتين أو البروتين سينسايز. طب الريست اوف جينوم is unaccounted. unaccounted يعني مش محسوب، يعني البارت الباقي من الجينوم unaccounted يعني أنا مش قادرة أحسبه، وده بيبقى عنده unknown function. يعني إيه unknown function؟ يعني أنا مش قادرة أحدد وظيفته إيه، بس هتعرف بعد كده إن طبعا زي بقية ال RNA هو بيساعد لو حصل دروب أو حصل أي damage في حاجة. Dear students, we described in this session the DNA and the genome structure. I would you enjoy our time today and you understand what I can say about the DNA structure. Thank you for watching. Goodbye.